So I'm going to talk to you about GentiBio. We're a company that's engineering Tregs, and we're really positioning ourselves to deliver on the promise of Treg therapeutics. We're developing this approach, and we have the goal to reestablish tolerance in patients suffering from autoimmunity, as well as in indications with autoinflammatory disorders, as well as allergic diseases. So far, we've built a very seasoned leadership team, as well as scientific founders with a deep experience in cell therapeutics. And I think importantly, we're really focusing on improving the Treg platform to address key bottlenecks that are associated with bringing Tregs to patients. And this includes the scaling, the phenotype quality, and the specificity of the Tregs. We're creating a modular approach that allows us to rapidly create different products. And we have a pipeline strategy, as you'll see, where we're looking to incrementally build value from product to product as we move from autologous into allogeneic indications. We have a clear sightline to the clinic, and we have a strong investor syndicate. Recently, this past August, we closed a $157 million Series A, and our lead program in T1D has just started IND-enabling studies. So as I mentioned, we're building value over time with our approaches. We have both autologous and allogeneic approaches that we're developing. So we're going to start with our first one or two indications in the autoimmune area, focusing on autologous approaches, patient-derived cells. And then I think importantly, we're going to move very quickly into first-generation allogeneic engineered Tregs. And we can do this in autoinflammatory diseases. And I think this is an amazing application of Treg therapeutics, where I think it really takes advantage of some of the key capabilities of Tregs to suppress inflammation and to promote tissue repair. And these are also indications that actually require an allogeneic approach. Ultimately, we're moving towards iPSC-derived technology, and so very interested in speaking with folks about that technology. So we think there's an incredible opportunity for Tregs to address a whole host of different diseases. And when I started at GentiBio, together with Kathy Thutt, we worked on a program to really establish what our pipeline would be, and we looked at really all the different dimensions, starting with scientific and moving to clinical and commercial assessments of these different indications. And based on that, we came up with this pipeline, which you can see here, focusing on the green. I won't go through the whole pipeline, just to focus on a couple. In T1D, we're focusing there, which is our first program going into early onset T1D in adults, quickly moving into children, then ultimately moving into patients that are at high risk for T1D. The other one I'd like to highlight, which is our first allogeneic program, is viral ARDS, or acute respiratory distress syndrome. Like I said, I think there's an incredible opportunity for Tregs in the autoinflammatory disease area, and this is certainly a high unmet need, not just due to COVID, as we're all aware, but also before that associated with influenza infections as well. So we have all this opportunity, but there's challenges. So we're building on a tremendous amount of work that's been done, obviously, in terms of great advances that have been made in terms of the biology and the understanding of Tregs and what they can do. They've been into the clinic, at least in a polyclonal adoptive transfer approach, non-engineered. And certainly we're building on the capabilities that have been built with CAR T therapies in terms of manufacturing. But Tregs have their own particular challenges, and I think this is the core of what GentiBio is really all about, is resolving these challenges. So certainly, if we start with Tregs and you try to fish them out, say from the blood from a patient as your starting material, these unengineered Tregs are rare. They're about 5% of the total CD4 population. And then they're heterogeneous. And in fact, they can potentially transdifferentiate back into T effectors. So they're both a rarity and potentially a stability challenge. So our approach has been really to start with more abundant cell types, like CD4 cells, and engineer them into Treg phenotypes, and really do it in a way that we can maintain stable FOXP3 expression, which is the master regulator for Tregs. A second one, a challenge, is that Tregs require IL-2, but they don't produce it themselves or rely on other cells to produce it. And so we've come at this challenge by creating a chimeric IL-2 signaling receptor. This allows us to turn this signaling on and off using a small molecule. So it gives us a great deal of specificity. It's only activating the signaling within our engineered Tregs, and we can tune it. We can tune the signaling up and down based on how much and how often we give the small molecule. Now, third challenge is antigen specificity, and I don't think there's too much disagreement about that. This aspect, pretty well recognized in the field, but certainly for Tregs, the specificity is actually quite important, as I'll describe, in terms of turning on key functions that are associated with their suppressive capabilities. So if you step back and you put this all together, you have this cell where now we have constitutively expressed stable FOXP3 expression. We have this chimeric IL-2 signaling receptor providing the IL-2 signaling support to the cell, and then we have TCR and CAR expression. And what we won't go into today, but we've created a very elegant and robust way to combine our chimeric IL-2 signaling for selection and gene editing approaches to have a very scalable and robust manufacturing process. So diving in a little bit deeper on each one of these platforms or pillars of our technology. So like I said, rather than start from these rare and heterogeneous cell types, we engineer T cells from more abundant, Tregs from more abundant cell sources. And so the way we do that is depicted in this diagram. We engineer, using gene editing approaches, a very strong MND promoter directly in front of the first codon of the FOXP3 gene. And that has two important effects. Number one, it converts those CD4 cells into a Treg phenotype, since FOXP3 is the master regulator of Treg biology. And then secondly, and very importantly, where we put that promoter, it's downstream of all the regulatory elements. So regulatory elements can lead to natural Tregs downregulating FOXP3. We avoid all that. We completely divorce it, and we can maintain a stable Treg phenotype by maintaining stable FOXP3 expression. So when we do that, when we start with bulk CD4 cells and engineer them into Tregs, you can see, number one, that we can provide robust activity that's just as effective in vivo as naturally occurring Tregs, and the expression profile of these genes is very similar to natural Tregs. So on the left, you can see in a GVHD model that when we induce GVHD, if you focus on the green line, you can see we can reduce and delay the onset of GVHD. And importantly, if you compare that to the blue line, it's just as effective as a naturally derived Treg. And then on the right, you can see an expression profile showing that we have a very similar expression profile to the naturally occurring Tregs. And this is all taken from a published paper from one of our co-founders, David Rawlings, in Science Translational Medicine 2020. So what about the second pillar, IL-2 signaling? Why is that important? Well, we think it's really quite important because of the fact that if you look at certain autoimmune diseases where you look at IL-2 signaling within regulatory T cells, there's a number of publications showing that this IL-2 signaling is defective. And in many cases, there can also be a scarcity of IL-2. And this leads to an imbalance between the Tregs to T effectors, both in terms of their functionality and in terms of their numbers. And so if you could supply this IL-2 signaling specifically, this gives us a way to really restore that correct balance. So the way we do this is we start with the IL-2 receptors that are involved in signaling, the IL-2 beta and gamma. They would normally bind to IL-2 and dimerize and create the signal. And so what we've done is replaced the extracellular domains that would bind to IL-2 and lead to the dimerization with 2 domains that dimerize in response to the small molecule drug rapamycin. So what this gives us now is a very highly selective way to turn on IL-2 signaling and do it in a tunable way. We can turn it up and down based on the frequency and the dose of rapamycin that we give. And so this has 2 very important functions. Number one, we can use this on a manufacturing side and a scalability side because this turns out to be a selectable marker. So only the cells that are edited get the IL-2 signal. So if we edit the cells, put them in rapamycin, we can selectively expand the edited cells in the rapamycin, and so we can have a very highly enriched product without the need for any sorting or additional purification technologies. Now, on the right shows how we can use that now in vivo, ultimately in the patient. Here we've dosed these Tregs into a mouse and then come in with increasing doses of rapamycin. And so what you can see very nicely in both the peripheral blood and the liver is that we get a nice dose-dependent increase in the level of engraftment of these cells. And we also have some data to showing that this can also increase the efficacy of our cells as well. So in both these ways, we think this is a really nice piece of technology that allows us to produce the cells, number one, and then also increase their potency and efficacy, number two. Moving on to tissue targeting, which is our third pillar. Certainly, it's important to get the cells where you want them, and the targeting gets you that. But the point of this slide is for regulatory T cells, having that signaling is critical for some of their key suppressive functions, namely bystander suppression and infectious tolerance. And so on the left, with bystander suppression, that's where that single Treg recognizing that single antigen can suppress T effectors that may be recognizing multiple different autoantigens. Obviously quite critical in diseases like T1D, multiple sclerosis, where there's a significant degree of antigen spreading. And then on the right, infectious tolerance is a mechanism whereby that Treg can actually recruit cells that may ultimately contribute to promoting the disease to cells that are contributing to suppressing the disease. So it's really through this amplification of mechanisms that the CARs and the TCRs get you that. And so I think this data really nicely demonstrates the point on the importance of the TCR. So this is data from our T1D program where we compared directly an islet-specific TCR within the context of one of our engineered Tregs to engineered Tregs that don't have an islet-specific TCR. And hopefully you can see quite dramatically is that the targeted islet-specific TCR Treg can nearly completely prevent the onset of T1D in this model. So certainly with all those mechanisms, then we can, and these pillars, we can put this together in products in a modular fashion that leads to our pipeline where, as I mentioned, our first program is in T1D. We're just starting IND-enabling studies. Our first allogeneic program, as I mentioned, is in viral ARDS. Very excited about that one and the potential for Tregs to treat autoinflammatory diseases. And then we're going to be determining a second autologous program. And we have a number of programs ongoing that are listed at the bottom, including IBD, multiple sclerosis, and a very interesting indication, primary biliary cholangitis, which is part of a cluster of 3 different autoimmune liver diseases. And so just to wrap up, I think our focus at GentiBio is to, as we say, make Tregs better or potentially best. We're doing this in a number of ways through establishing world-class leadership with deep experience in cell therapeutics. Importantly, building a platform we think really addresses the key bottlenecks that are associated with Treg therapeutics. And then having a strategy where we build commercial value of each product over time to really unlock the full potential of Tregs. So thank you for your attention, and I can take any questions in the back afterwards.